Hello everyone, in this lecture I will talk about the basic steps in plant breeding. So here is the outline of my presentation. First I will discuss to you the definition of plant breeding, and this will be followed by the topic on the basic steps in plant breeding. So what is plant breeding? Plant breeding is actually defined as the science, art and business of changing the genetic design of plants for human benefit. So you have seen this uh, figure before, wherein you have here the plant breeding surrounded by several relevant disciplines, and a plant breeder must have knowledge in each of those fields in order for him to master the field plant breeding. And we also discussed in the last lecture that the incorporation of science-based approaches has accelerated the crop improvement process because with all these scientific disciplines, it helped the plant breeder to understand biological process. And say, for example, uh, the plant breeder is able now to unravel the genes underlying the complex traits. Plant breeding is not only a science, but it's also an art. And as an art, it requires knowledge, skill, and judgment. We also have here selection, and remember, selection is an art of discriminating biological variation within a population to identify and choose the best ones. So, as a plant breeder, he must have a visual selection, or what we call breeder's eye, in order for him to easily identify variants. And if those variants are interesting and superior, then those variants will be advanced and will be evaluated for the next couple of seasons, years, and environment. We also have here agiotype breeding. So what is agiotype breeding? From the word agiotype, it's ideal type. So here you have a rice variety that is low tillering but high yielding. But in order for you to develop this, you at least have this, for example, tall conventional plant, and what you want here is to shorten the height first, but retain all the other characteristics, say for example high yield. Once you develop that genotype, then you now have here a shorter height but with several tillers. Now, in order for you to develop your ideal type, then you have to look in your germplasm for lines where it produces less tillers but also high yielding, and you will cross them, and later on you will develop this low tillering agiotype rice variety. Plant breeding is not only a science and it's not only an art, but it's also a business. Why? Because plant breeders are continually focusing their effort on developing new cultivars to meet the market demands. These new cultivars must have the desired characteristics or traits desired by the end users. You have heard also of seed companies. They actually invest their resources just to develop these cultivars, again, to meet the market demands. You have here a pie graph showing you the seed companies playing in the market. So we have here first the Bayer, 22%, Corteva, 19%, and all other seed companies. So there's not only one, but there are many seed companies in the market playing with each other, playing against each other just to sell their new varieties and encourage the end users to buy those new developed varieties. On the right, we have a figure showing us the real seed prices of selected vegetables. So this is uh, between 1990 until 2016. So as you noticed, between 1990 until 2005, there's not much increase on the prices of those seeds developed by seed companies. However, um, after 2005, you have a sudden spike or sudden increase on the prices of individual vegetables. So probably those seeds were now or undergone a process and developed with certain traits desired by different users. Say, for example, for the industry or for consumption, wherein those varieties contain the desired characteristics like high nutritional content or medicinal properties. That's why so there's a sudden increase in the price of those vegetable seeds. So here are the steps in plant breeding. This is the basic. but there could be many modifications. But here we have first, there should be a breeding goal. So your breeding goal must be, let's say for example, you want to breed for higher quality, higher yield, and resistant against biotic or tolerant to abiotic stresses. Now once you have formulated your breeding goal, you have to check your germplasm for variability. So we have here genetic variation for the desired traits that must be present in the germplasm, because here, yeah, after this you need to select, and before you can select there should be enough genetic variation. And of course, if there's no variation, then you have to create your variation by introducing new varieties or doing hybridization, or ask genetic material from gene banks or biorepositories, or maybe you can go to farmers field and ask for genotypes or varieties they maintain in order for you to add that to your germplasm to increase the genetic variability. Now, once you have confirmed that you have enough genetic variability for the trait of interest, then you can now go to the selection process. So here you can now perform the art of discrimination, and you can use your breeder's eye to select or identify the best variants. So you will do this in a couple of generations. Uh, Maybe you will reach up to a seventh generation just for you to be able to select the superior lines. So once you've selected the superior lines, you have to proceed to the next step, which is the evaluation of genotype. So superior genotypes are evaluated across different production environments. So here also the superior genotype must be compared to a check variety, and the superior genotype must have the DUS characteristic, so the distinct, uniform, and stable characteristics before it will be called a variety. And that superior genotype must also have superior characteristics over the check variety. So once you've done with the evaluation of the genotype, then you need to proceed to the certification and release of new variety. So here we have certifying agency to approve and register the new varieties you have just developed. And this is very important because once you have registered your variety, you can gain breeders' rights and you have protection against illegal release of your cultivar, and you now have ownership over your cultivar. Also, there are some advantages you can get once you have registered your variety before you can release it. So here are the steps in plant breeding. Again, breeding goals, creation of variation, selection process, evaluation of genotypes, and lastly, the certification and release of new varieties. So let us discuss each step in plant breeding. So let's discuss first the breeding goal. So whenever you formulate your breeding goal must be clear and SMART. So SMART stands for specific, measurable, attainable, realistic, and time-bound. Now, your breeding goal must also target a specific end user or group of end users. So later on I will discuss to you what are the groups of end users. Your breeding goal must not only to address the present needs, but also the future demands. So as a plant breeder, you have to think in advance. You can develop new lines or varieties, not only for today, but also for tomorrow. Okay, so again, your breeding goal must be clear, smart, and must target a specific end user, and should address not only the present circumstance, but also future demands. So here are your guide on formulating your breeding goal. So say for example, you want to target the world food supply, so you will have a breeding goal to develop varieties that are high yielding and early maturing. And then of course, if you have that, then you can help in the world food supply. Another target in your breeding goal is to improve the quality traits. Say for example, the nutritional content, the protein content, and other properties, qualities in a crop. Also, you can target breeding for resistance against biotic stresses like pests and diseases. Or you can create a variety that can adapt to various environmental conditions like saline or drought or water stress environment. Maybe you can also change the maturity of your plant. So from the, uh, you know, that could yield longer. So you want that to be shorter. So you have early maturing, and it's very important because also for food supply, if the earlier harvesting, it's the better. Also for photo insensitivity. So, say for example, you introduced. By the way, introduction. There will be a topic on that. So I will discuss that in the next couple of lessons. So, for example, you introduced a variety from Japan or a genotype from Japan, and of course we know Japan is a temperate region, and then they have different photo periods. So, in order for you to improve that introduced variety, then you have to develop, you have to improve that in order to cope up with the photo period here in the Philippines. And lastly is your breeding goal again is must met the end user demands. So remember, end user demands are very important because of course they are the end users. So what you are doing right now, those end users are benefiting. So here are the types of end users. We have producers, processor, and consumers. And then producers, they basically need high yielding, early maturing, and disease resistant cultivars. So these producers, they are interested on increasing their profit. That's why they need high yielding and early maturing varieties. So remember, early maturing, the earlier the better. So early maturing, early harvesting, early income. Of course, this is resistant because what is high yielding if those are disease susceptible. So whenever you plant them, it would be nice if they get high yield and resistance, so there will be increase in profit. Processors, on the other hand, they demand for cultivars as raw material to create new products. Say, for example, in the pulp industry or pulp and fiber industry, so abaca, you can breed abaca varieties that can produce higher yield of fiber and also with fiber strength. Or say, for example, in tomato paste, so you need to develop a variety of tomato that has uh, paste properties. And the third type of end user are the consumers. So this includes you, me, and I think everyone because we basically look for cultivars with excellent taste, flavorful, and of course nutritious, and with good appearance. So again, those are the three types of users. Again, producer, processor, and consumers. The second step in plant breeding after you have formulated your breeding goal is the creation of variation and population formation. So you have to check your germplasm. For example, you want to breed for resistance, then you have to check your germplasm if you have available genetic diversity for disease resistance. So if not, then you have to look for sources of genetic diversity. So for example, in the gene banks, those are biorepositories that preserve genetic material. They might have those lines that has resistance against specific biotic stress, or maybe you can go to farmer's field. You can collect land races, heirloom varieties. Heirloom, those are all varieties or traditional crops. So those are good sources of genetic diversity. If those sources are enough for you to increase the genetic diversity, then you're good to go. However, if you don't have that, then you can do mutation and hybridization. If possible, you can do biotechnology, but here in the Philippines, this is not advised yet. So maybe you can just go on the mutation. Then after that, then you can proceed with the population formation. You can do two-parent population, three-way population, four-way population, or even a complex population in order for you to increase your population for your selection process, which will be the next step. So the main purpose of this selection process is to identify, select, and advance potential genotypes. So here you will use your breeder's eye, and you will also incorporate here the selection methods. Later I will discuss to you the selection methods in a couple of lessons, specifically for self-pollinated, cross-pollinated, and clonally propagated crops. There will also be breeding techniques. You can do back crossing, you can include genetic sterility, hybrid seed production, etc. There are also like what we call traditional and modern breeding approaches. So you can choose traditional; those are non-GM approaches. But modern breeding approaches, this encompasses modern techniques that includes biotechniques, the use of um, GMO or the recombinant DNA technology to incorporate new genes or wild or genes from other species. So again, the third step in plant breeding is the selection process, mainly to identify, select, and advance potential genotype, which will be evaluated in a couple of years, seasons, and location, which will be in the fourth step in plant breeding. So here, your superior genotype selected during the several generation of selection may be used as parent in your breeding program or may be released as new variety. So a variety is a group of plants with a common set of characteristics. So a variety should have these distinct, uniform, and stable traits. So that defines a variety from a genotype. So here in the selection process, we term it genotype because it's not yet a variety. And once it passes this DUS test, then that will be the time that that genotype be called a variety. And in this DOS test, you will test this across the location, season, and years, of course, with, and I mean, you will compare that to a check variety. And if your superior genotype is comparable or even better than the check variety, then that will be the time that you can proceed with the seed certification and release. And then you will contact uh, the governing body responsible in the cultivar certification and release. Here in the Philippines, we have the NSIC, or the National Seed Industry Council. So the last step is cultivar certification and release. This is to maintain the genetic identity, purity, and quality standards of crop varieties. This is essential because um, here you can gain breeders' rights, you can gain authority over your developed variety. You'll be protected from illegal distribution and multiplication of your variety. And also, if you have breeders' rights, um, you have the right and you have the royalty if ever. So you will earn here protection and of course um, you will earn from your developed variety. Okay. And in the last step, this includes seed testing and cultivar multiplication. Seed testing, this is the accurate and prompt analysis of seed quality. So we have the NCTs, and National Cooperative Testing. So that's part of evaluation of genotype and also connected to this cultivar certification in the NSIC. And after you have registered your variety, you have done this seed testing and you have checked the quality, then you can now proceed with cultivar multiplication. And after that, then you can do the distribution process. So again, these are the basic steps in plant breeding. So I will review. We have here breeding goals, creation of variegation, selection process, evaluation of genotype, cultivar certification, and release.